Try to do the following questions and check your level of knowledge. Plant Pathology Model Exam for Exit Exam. Part 2. 41. Which physiological process is directly affected by vascular wilt pathogens? A. Seed germination. B. Transpiration. C. Water and nutrient transport. D. Flowering. Answer: C. Water and nutrient transport. 42. One major effect of viral infections in plants is: A. Enhanced cell division. B. Disruption of hormone balance and stunted growth. C. Improved water retention. D. Increased root biomass. Answer: B. Disruption of hormone balance and stunted growth. 43. Plant pathogens can cause a reduction in yield by: A. Accelerating seed development. B. Improving photosynthetic rates. C. Disrupting metabolic processes and energy flow. D. Enhancing respiration efficiency. Answer: C. Disrupting metabolic processes and energy flow. 44. How do root pathogens typically affect nutrient uptake in plants? A. By enlarging the roots to absorb more. B. By forming mutualistic relationships. C. By damaging root tissues and reducing absorption. D. By converting minerals into usable forms. Answer: C. By damaging root tissues and reducing absorption. 45. What is the first line of defense in plants against pathogens? A. Antibodies. B. Cell wall and cuticle. C. Enzymes. D. Phytoalexins. Answer: B. Cell wall and cuticle. 46. Which of the following is an example of a structural defense mechanism in plants? A. Phytoalexin production. B. Formation of lignin. C. Enzyme secretion. D. Hypersensitive response. Answer: B. Formation of lignin. 47. What are phytoalexins? A. Defensive proteins. B. Structural reinforcements. C. Antimicrobial compounds produced in response to infection. D. Insect hormones. Answer: C. Antimicrobial compounds produced in response to infection. 48. The hypersensitive response in plants involves: A. Increased photosynthesis. B. Rapid cell death at infection site to contain pathogens. C. Callus formation. D. Root elongation. Answer: B. Rapid cell death at infection site to contain pathogens. 49. Which hormone plays a key role in plant defense signaling? A. Auxin. B. Cytokinin. C. Salicylic acid. D. Gibberellin. Answer, C. Salicylic acid. 50. Which of the following is a pre-existing biochemical defense? A. Callose deposition. B. Phytoalexin synthesis. C. Phenolic compounds. D. Lignin formation. Answer, C. Phenolic compounds. 51. Systemic acquired resistance, SAR, in plants provides A. Localized immunity only. B. Short-term resistance. C. Whole plant, long-lasting resistance. D. Resistance only to nematodes. Answer, C. Whole plant, long-lasting resistance. 52. The enzyme chitinase helps in plant defense by A. Breaking down bacterial cell walls. B. Digesting fungal cell walls. C. Increasing leaf thickness. D. Synthesizing chlorophyll. Answer, B. Digesting fungal cell walls. 53. What role do stomata play in plant defense? A. They promote pathogen entry. B. They regulate light absorption. C. They close to prevent pathogen entry. D. They produce antimicrobial toxins. Answer, C. They close to prevent pathogen entry. 54. Which of the following is a plant's induced structural defense? A. Stomatal closure. B. Formation of cork layers or tyloses. C. Production of essential oils. D. Opening of guard cells. Answer, B. Formation of cork layers or tyloses. 55. What does the term resistance gene, R gene, refer to in plant genetics? A. A gene that increases plant yield. B. A gene that enhances drought tolerance. C. A gene in the plant that helps recognize specific pathogens and trigger defense. D. A gene that promotes flowering. Answer, C. A gene in the plant that helps recognize specific pathogens and trigger defense. 56. The gene for gene hypothesis describes the interaction between A. Two different plant species. B. A plant resistance gene and a pathogen avirulence gene. C. Genes controlling drought resistance. D. Genes involved in seed dispersal. Answer, B. A plant resistance gene and a pathogen avirulence gene. 57. In the gene for gene relationship, a disease occurs when A. Both R and AVR genes are present. B. The plant has an R gene and the pathogen lacks the corresponding AVR gene. C. The pathogen's AVR gene suppresses the R gene. D. The pathogen lacks the AVR gene and the plant lacks the R gene. Answer, D. The pathogen lacks the AVR gene and the plant lacks the R gene. 58. Horizontal, polygenic, resistance is characterized by A. Being effective against only one race of a pathogen. B. Being race-specific and easily overcome. C. Broad-spectrum and durable resistance controlled by multiple genes. D. Being controlled by a single dominant gene. Answer: C. Broad-spectrum and durable resistance controlled by multiple genes. 59. What type of resistance is typically more durable over time? A. Vertical resistance. B. Monogenic resistance. C. Horizontal resistance. D. Cytoplasmic resistance. Answer: C. Horizontal resistance. 60. Which of the following is not a method used to develop disease-resistant plants? A. Traditional breeding. B. Genetic engineering. C. Grafting. D. Gene silencing. Answer: C. Grafting. 61. What is a major risk of relying solely on single gene resistance? A. Increased mutation in the plant genome. B. Rapid breakdown of resistance due to pathogen evolution. C. Toxicity to beneficial insects. D. Reduced photosynthesis. Answer: B. Rapid breakdown of resistance due to pathogen evolution. 62. Quantitative trait loci, QTLs, in plant disease resistance refer to: A. Genes that control flower color. B. Regions in the genome associated with multiple small effects on resistance. C. Specific avirulence genes in pathogens. D. Nutritional traits in plants. Answer: B. Regions in the genome associated with multiple small effects on resistance. 63. Which tool is commonly used to identify disease resistance genes in plants? A. PCR and molecular markers. B. Grafting. C. Foliar spraying. D. Cross-pollination. Answer: A. PCR and molecular markers. 64. Durable resistance is best achieved by: A. Relying on one major resistance gene. B. Combining multiple resistance genes and breeding for horizontal resistance. C. Avoiding hybrid crops. D. Reducing genetic diversity. Answer: B. Combining multiple resistance genes and breeding for horizontal resistance. 65. Which of the following is not a major component of the plant disease triangle? A. Susceptible host. B. Virulent pathogen. C. Favorable environment. D. Soil pH. Answer: D. Soil pH. 66. The most favorable temperature for many fungal plant pathogens is usually in the range of: A. 0 to 10 degrees Celsius. B. 10 to 20 degrees Celsius. C. 20 to 30 degrees Celsius. D. 35 to 45 degrees Celsius. Answer, C. 20 to 30 degrees Celsius. 67. High relative humidity typically favors the development of A. Viral infections. B. Fungal diseases like downy mildew. C. Bacterial wilt. D. Root nematodes. Answer, B. Fungal diseases like downy mildew. 68. How does wind affect the epidemiology of plant diseases? A. It increases soil fertility. B. It limits disease spread. C. It helps disperse spores and vectors. D. It kills pathogens directly. Answer, C. It helps disperse spores and vectors. 69. A dense plant canopy can A. Increase light penetration and reduce disease. B. Decrease humidity and slow disease spread. C. Increase humidity and create favorable conditions for pathogens. D. Prevent vector movement. Answer, C. Increase humidity and create favorable conditions for pathogens. 70. Which factor directly affects the rate of disease spread in a population? A. Host resistance. B. Soil mineral content. C. Use of irrigation. D. Amount of rainfall. Answer, A. Host resistance. 71. The role of vectors, like insects, in plant disease epidemiology is to A. Control weeds. B. Improve pollination. C. Transmit pathogens from plant to plant. D. Increase water uptake. Answer, C. Transmit pathogens from plant to plant. 72. Monoculture cropping systems tend to A. Reduce disease incidence. B. Encourage biodiversity and disease resistance. C. Promote rapid disease epidemics. D. Eliminate pathogen populations. Answer, C. Promote rapid disease epidemics. 73. The time between infection and appearance of symptoms is known as A. Incubation period. B. Infection window. C. Pathogen latency. D. Disease triangle. Answer, A. Incubation period. 74. Which of the following abiotic factors most significantly influence disease development? A. Soil pH, temperature, and moisture. B. Vector population. C. Host genotype. D. Pathogen virulence. Answer, A. Soil pH, temperature, and moisture. 75. Which of the following is a cultural method of managing plant diseases? A. Spraying fungicides. B. Crop rotation. C. Using resistant varieties. D. Applying biocontrol agents. Answer, B. Crop rotation. 76. What is the main advantage of using resistant crop varieties? A. Increases the need for irrigation. B. Reduces dependency on chemical pesticides. C. Enhances flavor of produce. D. Makes plants more attractive to pollinators. Answer, B. Reduces dependency on chemical pesticides. 77. Which method involves the use of living organisms to control plant diseases? A. Chemical control. B. Physical control. C. Biological control. D. Quarantine. Answer, C. Biological control. 78. Trichoderma species are commonly used in plant disease management as A. Pathogens. B. Fertilizers. C. Biocontrol agents. D. Herbicides. Answer, C. Biocontrol agents. 79. A major limitation of chemical control is A. It is always too cheap. B. It enhances soil fertility. C. Pathogens can develop resistance over time. D. It increases crop genetic diversity. Answer, C. Pathogens can develop resistance over time. 80. The removal and destruction of infected plant debris is an example of A. Genetic control. B. Cultural control. C. Mechanical control. D. Biological control. Answer, B. Cultural control. Thanks for watching. Please subscribe our channel and share for your friends. See you next time.